All right, we're going to go over test 2 for spring 2014. If my pen writes here. All right. So as always, the unit number is listed here, and the specific slide is listed right next to it. So the first 12 are from Unit 4. Which reaction does not take place in brain cells under normal conditions? So it's good practice to go through all of these and figure out what reaction that actually is. But the answer is D. That's done by fructokinase, and that's the liver processing fructose. So if you see fructose or mannose, galactose, chances are it's going to be a liver reaction. Everything else here looks like it's either from glycolysis or the Krebs cycle. So make sure you can name all of those enzymes as well. The protein is not found in muscle cells. Lactalbumin is found in the mammary glands. Remember, GLUT1 and 3 are the ones that are in all cells, so those GLUT families are going to keep coming back on every test. Pour any cell with a mitochondria. Lactate dehydrogenase, red blood cells and muscles. Hexokinase, all cells. Anything from glycolysis, all cells. Which molecule is essential for the synthesis of lactose and sucrose? So if you're looking at the pathways, the answer is UTP. A lot of these two are galactose and lactose only, but sucrose, remember, it's a glucose and a fructose molecule. So galactose will have nothing to do with the processing of sucrose. Which enzyme is not used in the conversion of fructose to pyruvate in the liver? So this slide keeps coming up. Make sure you know the difference between how fructose is processed in the liver versus in the adipose tissue. The answer to this one is aldolase. Remember, in the liver it comes in around step 5 or 6 of glycolysis, so it skips that first lyase here. Which enzyme is not used in the glycolytic oxidation of mannose, galactose, or glucose? The answer to that is triose kinase. That was used for the fructose pathway, and that's the only time we're going to see that enzyme in Biochem 2. Which metabolite is not synthesized in brain cells under normal conditions? Hopefully you could have answered that during test 1. Lactate is the red blood cell muscle under vigorous exercise conditions. I feel like I've said that 100 times in these videos. It's a point he likes to drive home. Which molecule is known to bind to galactosyl transferase? It's the alpha-lactalbumin again. Remember, lactose synthase is a multifunctional enzyme or a multi-part enzyme. It's made of galactosyl transferase and alpha-lactalbumin. So I'm looking at the first page. It looks like slides 7, 8, and 15 had pretty much all of the questions on it. So if you're just picking a few, go for those few slides. Question about the regulation of lactose. Which hormone inhibits? It's progesterone. Prolactin is going to be what's stimulating it. Prolactin stimulates the synthesis of lactose. All these other ones have nothing to do with lactose. Which molecule does not appear to be involved in increasing the risk of galactosemia? Remember, that's a disorder in the breakdown of galactose. It can happen with any of the enzymes. So those are not the answer there. So we have sucrose, which is fructose and glucose. We have lactose, which has a galactose in it. So the breakdown of lactose can result in the buildup of galactose and increase the risk. So the answer is sucrose. Quick note on this one, some of these are worse than others, or some are screened at birth. That's another thing he likes to ask about this disorder. So that's definitely going to be a question. Make sure you know that one pretty well. The net ATP yield of converting 1 molecule of mannose to pyruvate in the liver. This is on the very last slide of Unit 4. We're talking about net yield. So remember, with glucose, we get 4, but we invested 2, so the net yield was 2. And that's true for mannose, fructose, galactose. So the answer is 2 ATP molecules. This reaction does not take place in liver cells. Make sure you know what all these reactions are. The one that makes no sense here is E. Lactose plus water equals glucose plus fructose. Unless I'm missing something big, I don't think that's a real reaction. Which enzyme is known to be a complex protein? Lactose synthase. It's made of the galactosyltransferase and the alpha-lactalbumin. Everything else is just a regular enzyme. Which molecule is not typically found in brain cells? Only glucose is the only carbohydrate, or simple carbohydrate, that should be getting to the brain. The other one he likes to drive home with the brain cells is fatty acids, usually palmitate, or any sort of fatty acid should not typically be found in the brain. Which functional enzymatic activity of the PDH complex is pantothenate dependent? So this is memorizing E1, 2, and 3 of the PDH complex and what vitamins, what co-substrates, and prosthetic groups are involved. So that's all on slide 29. The answer to this is E2. Pantothenate, vitamin B5, is involved in any time you see a reaction with CoA in it. That's your signal that those go together. These molecules has the lowest number of charges at physiological conditions. So that's a protein, 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 protein. These all being enzymes and this being a transporter. So succinyl-CoA is the only metabolite there. So that's the answer. Without knowing anything about it, enzymes and proteins are always going to have many more charges than any of the metabolites, even 1,3-BPG with a -4 charge. Which condition inhibits the PDH complex Krebs cycle rate and oxidative phosphorylation? The answer is high energy charge. The whole purpose of this is to make ATP, which is high energy. So high energy charge is going to inhibit it via negative feedback. Multifunctional enzyme, alpha-ketoglutarate dehydrogenase, same as the PDH complex. So PDH could be another answer if he just switches one. Mitochondrial metabolite is generated by a condensation reaction. That's a tough question. I wouldn't have known that one. On slide 44, it lists all the reactions, and rather than seeing them by the usual enzyme class, it gives a couple different reaction types. So if you look through there, the answer is citrate. So if you've studied everything else, then you might want to go for that, but I would consider that kind of a secondary priority. Like I said, and I think in the last test, was look for all the reactions that involve water. That's another popular question if you're not memorizing the full reactions and reaction types. Flavin enzyme, succinate dehydrogenase. Anytime you see FAD, FMN, where that F is flavin. And this dehydrogenase is the one that makes the FADH2. Which metabolite is not a net product of the TCA cycle in brain cells? Acetyl-CoA is not a net product. It's a reactant. It goes into the Krebs cycle. All of these other things come out of it. Which enzyme is a lyase? It's fumarase. Even though it has water in the reaction, it's still going from 1 to 2 or 2 to 1. It's the key indicator of the lyase. Which molecule stimulates the conversion of pyruvate to acetyl-CoA? So going from pyruvate to acetyl-CoA, we're producing NADH as one of our products. So if you have extra NAD+, that's going to stimulate the reaction to happen. I know acetyl-CoA and ATP inhibit this reaction. I'm not sure if he mentions anything about palmitate. How many carbons of fructose are fully oxidized by enzymes of the Krebs cycle? So fructose, 6 carbons in it. So we have 6 carbons, and if this could be glucose, mannose, it doesn't matter. Fructose is just there to confuse you. So we're going to break it down into 2 pyruvates, which each have 3 carbons. And then when pyruvate enters the PDH complex, 1 carbon is going to be oxidized by the PDH complex, and 2 are going to be oxidized by the Krebs cycle. So that happens with both pyruvates, 1 from the PDH, 2 from the Krebs cycle. So 2 plus 2 is 4. If you were to change this to PDH, the answer would be 2. If you were to say oxidized by glycolysis, the answer would be none. If you were to say oxidized in the mitochondria, the answer would be 6. It's a good example of where you need to be able to track the full oxidation. When you're looking at full oxidation, you're thinking CO2 as a product. So you get a CO2 from the PDH, and you get 2 CO2s released in the Krebs cycle. So make sure to know slide 54 really well. It's towards the end. For precursors, this one is talking about purines and pyrimidines. The answer is oxaloacetate. The thing is pyrimidines that alpha-ketoglutarate does. I know this only does one of them, and oxaloacetate does both of them. This vitamin does not appear to be essential from alpha-ketoglutarate to succinyl-CoA. So that's the same as the PDH complex, which was vitamins B1, 2, 3, and 5. 1, 2, 3, and 5. Folate's the answer. Krebs cycle metabolite is the most immediate precursor of glucose. The answer to that is oxaloacetate. It's again on that slide 54, so make sure to memorize that one really well. All of the anabolic pathways. All right, on to Unit 6. Which functional enzyme is known to be membrane-bound? This is the physical link from the Krebs cycle to oxidative phosphorylation. Succinate dehydrogenase is the membrane-bound one. The most important things to know about that. Which molecule is inhibited by amytal? It's complex 1. So slide 90 kind of summarizes all the regulation. You should have that one memorized very well. It's going to come up a couple more times, and it will in every test. Enzyme is found in the cytosol and in the mitochondrial matrix of cells. Aspartate aminotransferase. So make sure to know the shuttles really well and what things happen on the cytosol side versus the mitochondria side. And if any are membrane bound there. Which of these ETC molecules has the lowest standard reduction potential? This is the one I don't really know how to explain it. I think I had all of these listed in the video as the ones I knew, as having relatively higher ones, and I don't think copper was on there. So no, copper was the answer for that. There's a chart on slide 12. Molecule is not a product of oxidative phosphorylation. Succinate is a reactant, not a product. Everything else here is created at some point. So this reaction takes place in complex 2. You know, complex 2 is the one with the FADH2 to FAD. So without knowing the reactions at all, just by knowing FAD is involved in complex 2, that's the loose association you need to answer that one. All right, last page. Completely oxidize a molecule of glucose using glycolysis, PDH, Krebs, ETC, and the MAS shuttle. There's our first useful piece of information. MAS has 32 ATP coming from it. We want to know what fraction is generated inside the mitochondria. So first, know that the MAS shuttle, the MAS shuttle, generates MAS ATP versus GPS, which only has 30. So you can calculate where all these are coming from, or you know that 2 are coming from the cytosol, a net 2 of ATP, and then everything else is coming from the mitochondria, PDH and beyond. So 32 minus 2 equals 30. 30 mitochondria divided by a total of 32 is going to give you 93 point something. So the answer to that is 94. So for these questions, make sure you know 2 are coming from glycolysis and know the shuttles, and then he's going to ask either mitochondria, he might ask Krebs cycle in particular, he might ask cytosol. So we know 2 came from glycolysis, 5 came from the PDH, 20 came from the TCA cycle. molecule has not yet been extracted from the inner mitochondrial membrane? I'm really not sure how to answer that one. I know in like various slides we've seen these other membrane-bound, mitochondrial membrane-bound enzymes and transporters come up, but we've never seen the oxaloacetate carrier. So that's the answer. We've talked about ATP synthase. This is in the ETC. Pyruvate translocase was mentioned in getting it into the mitochondria, and thermogenin is shown on, I think, slide 86 or 7 to be a membrane-bound protein. Which reaction is catalyzed by superoxide dimutase? Answer to that is E. Should have all those reactions down on slide 38 for this one question. Which protein is not a component of the MAS shuttle? Shuttles are summarized on slide 68, and this is part of the GPS shuttle. Molecule does not appear to participate in thermogenesis. So it's summarized in 86 and 87. ATP synthase is inhibited. It's uncoupled, so the electrons can't reach this. But this is thermogenin, UCP1. Fatty acids are being burned for fuel during this. And it starts off the electron transport chain, but is uncoupled before it gets to synthase. So that's the reaction. Risk of this disease does not seem to be increased by ROS molecules. Osteoporosis, I've never seen this question before, but there's a slide on 42 that summarizes ROS related diseases. Enzyme is copper and zinc dependent. That's the superoxide dimutase. Physiological uncoupler of oxidative phosphorylation. That's thermogenin, or UCP1. And that's the end of the test. So good luck.